So good afternoon, everybody. So my name's John Bowman. I actually work I'm in the University of Tasmania based in Launceston. I'm actually in the Tasmanian Institute of Agriculture. So me doing Tasmanian Devil sort of puzzled my boss a little bit at the time, because I've worked in many different other areas of microbiology, food production systems, food safety, previously Antarctic microbiology. But I thought there was this amazing opportunity to actually study microbiology of course, what is, a very iconic species, which is, classic to Tasmania, perfect place-based research. So this research is essentially exploratory and is focused on looking at the microbiology of essentially bite wounds and the tumour cells, or the tumour, basically, collected from wild animals or wild devils. And the idea is that, of course, devils are very social animals, and they tend to be rather aggressive in terms of their socialization. So they tend to bite each other quite a bit. So this creates an opportunity for infectious diseases to actually occur in the presence of the DFT tumours. As Ruth was mentioning, and also Andy, the tumours do create a situation where there is a increased susceptibility to secondary infections because of the MHCC downregulation, which I don't pretend to understand very much, but I've certainly got a bit of an education now. And there's been some hypothesis that maybe there could be certain antimicrobial treatments could improve health outcomes for devils. So based upon some studies, mainly to do with the oral cavity type related microorganisms. So, and also there wasn't very much data specifically for these sites. There's plenty of other data for like gut microbiome, for example, in the Tasmanian devil, but not so much directly these sort of more skin surface sort of locations. And so the idea was that we wanted to ask the question, are certain bacteria enriched in the tumours and the bite wounds that might be interesting to understand? Because it's possible, are these organisms also present more broadly within the animal population and even maybe intersecting with humans? So that's sort of like the One Health connection. It might be a little bit tenuous, but it's a developing story, let's just say that. Now, I just wanted to say one thing. You've got to be a little bit careful when it comes to cancer microbiomes. Very controversial, because I believe that at one point people were very, very enthusiastic about cancers have their own sort of sets of microbes, and then we can diagnose them, and maybe we could use that information to cure cancer maybe. But of course, it was all discovered that it was not actually real. It was misinterpretation of data. And the technology is, particularly in microbiology, like in metagenomics, and just understanding microbes and how they're related to host systems, is just dramatically improved in the last decade or so. That our knowledge, our databases are still growing very quickly indeed. So, and genomes are just pouring into databases. So we just want to be careful and just say we're interpreting our results that actually they're meaningful. So having the One Health connection probably creates a bit more dimensionality to that in terms of like how we can actually interpret this information. So for this project, the work was done by Robin Taylor, who actually lives in the Highlands, so she probably experienced lots of snow in just recent days. And she, with a team of volunteers, sampled basically, animals in the wild, devils in the wild from several locations. And they sampled about 200 individuals in the locations indicated in that little image there. Now, 113 DFTD tumours were sampled, but they're not 113 individuals, because some of the animals had different locations on their face, like around their mouth. So it was actually equivalent to around about 50 or 60 individuals that were actually sampled. So there were lots of negatives and quite a few that were probably maybe a little bit hard to analyse in terms of like, were they positive or negative? But that probably includes the whole gamut of age classes, genders, through the year, like there's different seasons. So I think it was a pretty good sampling of the overall diversity within S. harrisii. This is just methods. I'm not going to bore you with that, but I just wanted to point out that when we're doing this type of sampling, even though we're just using swabs, we basically uncovered there's a very high level of microbial diversity present, and we're just looking at bacteria. We're not looking at higher life forms like, for example, protozoa or other things that might be there as well, fungi. So I had to constrain it a little bit to make it a bit more analysable in order to be able to do differential abundance analysis, which is one of these rather contentious methodologies because there's about 40 or 50 ways people do it. And then people argue the data is compositional or it's proportional and this sort of stuff. So the statistics are a bit nightmarish. So it's based upon a sort of a multiple methods, essentially, a relatively conservative analysis. So I'll just get to the results. So this is just the profiles from different sites of the animal, basically clustered. And you might notice the tumours and the bites clustered together with teeth and oral cavity samples, which sort of makes sense because you're assuming that microbes living in the mouth on the teeth and they're sort of biting will transfer to those sites. So there's interchange of microbes. Microbes are getting everywhere. Bacteria are very, very mobile. And we had also a lot of controls to control for the fact that Tasmanian devils are not exactly the cleanest animal in the world. They're rolling around in the soil and this sort of stuff. And but you can notice that in the other sites, like pouch jowls, underarm, that they're sort of distinctly a little bit different. I mean, there's lots and lots of overlap because you have to remember the microbes are sort of just spreading all over the place. It's not like there's distinct populations. It just doesn't happen with microbiology with that. So the idea is that we wanted to understand what are the bacteria that basically were being enriched in the tumours, as simple as that, and I'll go straight to the result. We found three that I would call robustly, believably, were being enriched. All three are pathogens, and one of them totally makes sense. In fact, there's already evidence that it's actually a very common organism associated with Tasmanian devil, and that's the third one, which is called Pasteurella multicida. So you can see that it's quite enriched in bite wounds. And of course, this is a good result because it's probably not, it's a bit of a logical conclusion because it's an organism that is a zoonotic pathogen that is spread by bite wounds. And it's even, Tasmanian devils even cause people to get, I think there was a case in 1992 where someone got bitten by Tasmanian devil and actually they got infected with Pasteurella multicida. Now, the other two were interesting, and I'll talk more about those in the next slides. So we'll just talk about Pasteurella multicida. This just shows the distribution in the different samples. It's based upon centered log ratios. It's just a way of normalizing the data and quantifying it in some way that doesn't make you go crazy. And what I think some of the interesting points is that this does occur. This organism is very widespread within the animal populations globally. It causes a whole range of diseases that infects humans as well. And it's also known to cause a disease in avians or particularly poultry called avian pasteurellosis, which in the acute form is about 40 to 50% fatal. Now, one of the interesting results, some recent paper, they used cell lysates within a tissue culture with feline fibrosarcoma cells and actually found there was and it actually increased the growth of this cancer. They believed it was linked to these proteins called MMPs, they're matrix metalloproteinases, which is to do with tissue growth. So this made me ask the question, what could be the Pasteurella multicida doing within the devil? Maybe it doesn't affect the cancer at all, but it's an interesting organism overall and could be further studied, I think. Just to go quickly through the two pathogens which were associated with the tumours. One is called Fusobacterium necrophorum, a fastidious gram-negative obligate anaerobe, which is also known to be zoonotic, causing soft tissue diseases, in particular one called Lemierre's syndrome, which is basically an infection you get in the jugular vein, which then causes blood clots, thrombocytopenia, which is something you might want to avoid. It's an emerging pathogen. So it's been found that Fusobacterium is a very substantial part of the devil gut microbiome. So there's a very high level diversity of this particular genus associated with devils, whether it's something to do with its diet and its lifestyle. There's many novel species, which I could see in the data. So this could be, as I said, further examined. Just basics to look at, I guess, maybe trying to get a better concept of the health of devils, maybe develop therapeutics or something like that. Maybe it's hard to say. It's really just an observation. So another one is just, one might say it's relatively similar in terms of like its overall concept. It also causes Lemierre's syndrome. It's an organism called Bacteroides pyogenes. It's a completely unrelated organism to Fusobacterium necrophorum. It's in a different phylum. But this one is also found to be frequently associated with basically dogs and cats. So in other words, people getting bitten. So these organisms are likely present within the just naturally circulating within in Tasmania and worldwide, I suspect, in terms of like the impact on wildlife as well as perhaps maybe also human health. Where it's actually going to go in terms of trying to predict trajectories of the significance of this is hard to say. I think this is basically just observations at the moment. But next steps could be, for example, to get isolates. This is just basically microbiome analysis. It's like a sort of the first step. Sequence the genomes and look for things like virulence factors, antimicrobial resistance. So partial multicide has been shown to have some resistance in Tasmanian devils, mostly to macrolides and third generation cephalosporins. But essentially, it's probably there's a lot of variability amongst that species. So another possibility is also to look at susceptibility to naturally produce antimicrobial peptides produced by the devil. It's cathelicidins, which is whether these organisms are actually naturally resistant. So that's it, really. Thank you very much for your attention. And we'll see where this research might go, but the Tasmanian devils hopefully are here to stay, and the microbes are probably going to be here to stay as well. So, okay, thank you very much.